Thank you for that nice introduction, Ben, and good afternoon, everyone. In my PhD project, as already mentioned before, in the University of Kiel, I work on high-dose inhaled antibiotic therapy. In this study that I want to present to you, I investigated the performance of rifampicin soft pellets in the twister inhalation device with or without sweeper crystals. So why is high-dose inhaled antibiotic therapy important at all? Let me explain that by the example of tuberculosis. Then inhaled antibiotic administration is compared with the standard oral therapy. Significantly higher drug concentrations can be found in the lungs. And these higher drug concentrations at the primary site of infection have different benefits. For example, the long treatment duration and the high dosing frequency could be reduced, and furthermore, by avoiding subtherapeutic drug levels, the development of multi-drug resistant tuberculosis can be prevented. In addition, the lower systemic concentrations lead to a lower probability of systemic side effects, making the therapy for the patient more comfortable and increase their compliance. To deliver the necessary high amounts of antibiotics in the milligram range into the lungs, the method of choice is dry powder inhalation. One option to formulate a powder that should be inhaled is the formulation of soft pellets. These soft pellets can be produced by controlled agglomeration of the micronized material. They consist of pure API without any excipients and have a spherical morphology with smooth surfaces. While inhalation, they deagglomerate into the inhalable primary particles again. In this study, a production process based on vibration was applied. In the first step of production, the micronized API is sieved through a 500 micrometer sieve and falls through a funnel onto an aluminum chute. This aluminum chute is set into vibration with an audio speaker connected to a stereo amplifier controlled by computer software. The soft pellets are formed by sliding down the aluminum chute. The sliding procedure was repeated for 3 times in this study. Afterwards, the soft pellets can be collected in a pen at the end of the slide. The benefits of the soft pellets are increased flowability, good redispersion, and reduced dust formation compared to the micronized material. And the fast and continuous production method itself needs no further solvents like in spray drying. For aerodynamic assessment, either 5, 10, or 20 milligram of rifampicin soft pellets were filled into 3 capsules each, or the sum of the dose was filled into 1 capsule. These capsules were loaded in the twister inhalation device from Aptar Pharma. When 3 capsules were applied, 1 capsule after another was loaded into the inhaler. For aerodynamic assessment, the fast screening impactor was used. And we observed that the emitted dose increases with increasing filling mass of the capsules. This is because of surface saturation inside of the inhaler. In contrary to that, the fine particle fraction decreases with increasing filling mass of the capsules, and as expected, the fine particle dose becomes higher when more powder is loaded into the inhaler. In addition, it was observed that the device performance is decreased when more powder or when the whole dose is loaded into 1 capsule instead of splitting it into 3 capsules. So for example, when 1 capsule is loaded with 60 milligrams of formulation, the fine particle dose generated is 50 milligram instead of 20 milligram. Then 3 capsules were filled with 20 milligram of formulation. This can be explained by a reduction of device resistance. When more powder is loaded into the inhaler, the airflow can't be converted into kinetic energy so efficient, and that's why the powder deagglomeration decreases. As already mentioned before, in high-dose inhalation, a lot of API adheres to the inner surfaces of the inhaler, and it has been described in literature that by the addition of large lactose particles, these API particles can be detached and the emitted dose can be increased. They call these large lactose particles sweeper crystals. So in the next step, 3 capsules were filled with 20 milligram of rifampicin soft pellets each, and different amounts of Inalac 70 and Inalac 120 from Megler were added as sweeper crystals. For aerodynamic assessment, the same experimental setup as described before was used. When having a look on the emitted dose, maximum can be found after the addition of 10 milligram inhalac 70. So the emitted dose increased from 65% for the pure soft pellet formulation to 74% after the addition of 10 milligram inhalac 70. This corresponds to an increase of 9%. The addition of inhalac 120 had no significant effects on the emitted dose. When having a look on a fine particle fraction and fine particle dose, again, a maximum can be found after the addition of 10 milligram inhalac 70. So the fine particle dose increased from 20 milligram for the pure soft pellet formulation to 24 milligram. This corresponds to an increase of 4 milligram, or as in relative value 20%. The addition of inhalac 120 had no significant effects on fine particle fraction or fine particle dose. Let me explain that observation. Inhalac 70 has, because of its larger diameter compared to inhalac 120, heavier particles, and therefore the inertia forces inside of the inhaler generate larger rotation radius compared to inhalac 120. This increases the probability of impacting particles on the inner walls, and therefore more API particles can be detached and the emitted dose increases. So it can be concluded that the emitted dose and the fine particle dose can be increased by the addition of inhalac 70. It would be interesting for further studies if alternative shaped particles or even larger particles have even better effects on the aerodynamic performance. So thank you for joining my presentation, and thank you for your attention. And I also want to thank my colleagues and my supervisor, Regina Scherlis, for supporting me. And now I'm looking forward to your questions.
Excellent. Thank you very much. We do have some questions for you. You said that you may extend your studies by looking at other shaped particles. Do you have a hypothesis about why particle shape may be important?
Sorry, I didn't get that.
Yeah, sorry. You said that you may look at different shapes of the sweeper particles in the future. A hypothesis why that may be important, why shape may have effect?
I guess that the surface of a particle that should be used for or as a sweeper crystal is important for the aerodynamic behavior inside of the inhaler. It would be really nice if the sweeper crystal would be large enough that you don't leave the inhaler, so that you don't cause cough in the patient if it would be used in a patient. Of course, the particles that I've used in this study were only lactose, because I don't wanted to investigate the shape of the particle itself. I only wanted to investigate how lactose particles perform in different sizes. And that's why I used inhalac 70 and inhalac 120. But I don't have any idea how different shapes of particles would behave. But I guess there will be an influence of the particle shape, of course. Can I answer the question?
Yeah. Let's move on to the next question, which is how specific do you think your findings are to your API? Do you think it would be different with different drugs?
Yes, of course. I think it would be different for different drugs. I also investigated the performance of isoniazid, but not with sweeper crystals until yet. And the performance of isoniazid isn't so good like rifampicin. So it underperforms rifampicin. But I guess this has other reasons than the adherence to the inner surfaces of the inhaler. I guess the isoniazid has more cohesion between the particles than rifampicin.
Okay, a final question. When would you consider the formulation to be optimized, and what criteria would you apply to determine that?
So you mean when I would say that the formulation is the optimum is reached? Did I understand that right? Okay.
Yeah.
I found a study where they investigated the inhalation of rifampicin and isoniazid, and they carried out the experiments with 20 milligram of rifampicin. So I would say that the optimum is reached when a fine particle dose of 20 milligrams can be generated of rifampicin. So the optimum is already reached when I use 3 capsules filled with a 20 milligram of formulation. I can generate with this method a 20 milligram fine particle dose, but it would be much nicer than the patient had not to inhale so much powder. So if I can reduce the amount of powder that should be inhaled, it would be better for a patient, because in high dose inhalation, the most observed side effect is cough because of the high powder amount.
Okay, thank you. We have a few more questions, but we're out of time.
Okay. Thank you, Ben.
Thank you very much to you, and thank you very much to all the speakers for their excellent presentations today and a fascinating session.